Hello friends, welcome to Bunk once again, and today we are going to discuss about the excretory system. It's our part 6 video, and we are going to learn about the physiology of urine formation. Okay, so in physiology of urine formation, basically there are three steps for it. So three steps. First one is ultrafiltration. Okay, second one is selective reabsorption. You can see filtration is what? Ultrafiltration. Okay, why we call it ultrafiltration, we are going to discuss this later on. And second one is selective reabsorption. It's not just reabsorption, but it's selective reabsorption. Like you have studied in selective permeable membrane, तो वो वही लेके जाएगा जो उसे लेके जाना है. So that there's a selection of substances also. Okay, so it's a selective reabsorption. Okay, and third one is tubular secretion. So this secretion is just not secretion, but it's tubular secretion. So as we are discussing about the physiology of urine formation, we should understand what is the physiology, how this urine is formed. We already discussed like the ammonia is getting converted into urea by the help of ornithine cycle inside the liver. Okay, so liver is the one which is performing the conversion of ammonia into urea. Why it is done so? Because urea is less toxic and that should be like ammonia is more toxic, so we cannot give directly into blood. So instead of giving directly into blood, what is done? It is first converted into urea and then it is given to blood. Okay, so पहले अमोनिया को यूरिया में बदला जाता है in with the help of ornithine cycle and liver, and then it is excreted out. So blood अब blood filter हो रहा है. Okay, so blood filter, that afferent arteriole is what? Afferent arteriole is called afferent because it is bringing the blood towards the nephron. Okay, so it's afferent, and efferent is like it is taking away. Okay, taking away the blood. So because the diameter of afferent artery, you can admire it's more as compared to the efferent one. So the diameter difference will create a pressure here in the glomerular region. Okay, in this glomerular capillaries, you can find a pressure. Why? Because the diameter of afferent arteriole is very much as compared to the efferent arteriole. So the pressure is building and blood is getting filtered. Okay, so blood is getting filtered. What all things are getting filtered? We will just discuss in ultrafiltration. Okay, so ultrafiltration, we understand what is ultrafiltration. Ultrafiltration is a process in which the pressure is created over the glomerular capillaries because of the difference of the afferent arteriole and efferent arteriole. Okay, so this one, because of this difference, a pressure will be created in this region. You can admire this glomerular capillaries and pressure is getting created and then blood is filtered. So the first one you can admire here, this first one is ultrafiltration. Okay, in ultrafiltration, what we know, ultrafiltration is a process of creating a pressure over glomerular capillary. Okay, so you see how ultrafiltration is done. Let us have a look in it. So suppose this one is Bowman capsule and this one is endothelium. I am making this a Bowman capsule like this. Just like this, and we will create a pedicel. Okay, so it's getting invaginating inside the blood capillary. So this one is blood capillary. Okay, this is BC, blood capillary. You can say glomerular blood capillary as well. It is glomerular blood capillary. Okay, so and the lining of blood vessel is what? The lining of blood vessel is called endothelium. So we can admire this endothelium. So endothelium is lining of any blood vessel. Since blood glomerular capillary is also again a blood vessel, so any blood vessel lining is called endothelium. So we can write here it's a lining of blood vessel. Now there is the invagination coming from Bowman capsule and going deep inside this region of, you can see this region of blood capillaries. Okay, so invagination is coming here like this and going deep inside the region of this blood capillaries. Okay, so this one is called pedicel. Okay, so this is the pedicel. So pedicel is what it is a invagination of Bowman capsule. This one is Bowman capsule. Okay, so what is pedicel? Pedicel is the invagination which is going deep inside the blood capillaries. Okay, and the distance between two pedicel you can admire here to here. This is slit pore. So slit pore is a thing through which blood is getting filtered. Okay, one more thing we need to acknowledge here. So there is a fenestra. Okay, so several calcified things you can find here. So this one is fenestra. So it is found in blood capillaries. Of course, the glomerular ones, which is called glomerular blood capillaries, and this one is fenestra. So what is fenestra? This is basically it is inhibiting RBC to get filtered. Okay, so it inhibits RBC, or you can say prevents RBC. So you can say corpuscles. Okay, all type of corpuscles, whether RBC, WBC, or platelets. Okay, so it inhibits corpuscles get filtered. Okay, so corpuscles are not getting filtered because of the fenestra thing, which is found in which is found in glomerular blood capillaries. Okay, so corpuscles to get filtered. Now this one here, this one is blood capillaries and this one is Bowman capsule. So anything in epithelium we have studied that there's a basement membrane, okay, and over the basement membrane anything is resting. So any type of tissue is resting over this basement membrane. So when endothelium is resting, it is again resting over the basement membrane, okay. And when the squamous epithelium of Bowman capsule is resting, it is again resting over the basement membrane. So the basement membrane of this endothelium would be like this, okay, and the basement membrane of this Bowman capsule squamous epithelium would be like this. So both the basement membrane lying towards each other. So this part we can acknowledge it is called BM or basement membrane. Okay. So basement membrane is negatively charged. It is negatively charged. Okay. So blood inside the capillaries, it is having blood protein also, plasma protein. So there are several plasma protein which is positive, which is again negatively charged. Okay. So what this basement membrane is doing? This basement membrane is it prevents the, what they are preventing? They are preventing protein structure, like protein, plasma protein to get filtered. Plasma protein to get filtered. Okay. So this is how basement membrane is preventing the plasma protein to get filtered, and this is how fenestra is preventing the corpuscles to get filtered. So what else will be getting filtered from this region? So this filtration will be glomerular filtrate. So GF is what? GF is glomerular filtrate. Okay, GS is glomerular filtrate, and this is the slit pore where there's pedicel stalk-like structure you can see, and the part where there is no pedicel, this is slit pore, and here in Bowman capsule, several intricately arranged cells are found. These cells are podocytes. Okay, so these cells are podocytes. It lacks at slit pore, so that blood can be filtered. Okay, means It is all over the squamous epithelium of Bowman capsule, but where does it lack? It lacks at the region of slit pore. Okay, so at slit pore there is no pedicel and no intricately arranged podocytes. So podocytes are what? Podocytes are intricately arranged. Okay, podocytes are intricately arranged cells in Bowman capsule. Okay, so this is how we can admire the podocytes here, which is arranged in an intricate manner inside the Bowman capsule, but they are not there in the slit pore, so that blood can be filtered and given as glomerular filtering. Okay, I hope you understand how ultrafiltration is occurring at the level of Bowman capsule and endothelium. So endothelium is the lining of blood vessels over the blood capillaries, and this one is Bowman capsule. So first thing what we need to know about ultrafiltration thing is ultrafiltration. Why ultrafiltration is called ultrafiltration? Because the diameter, okay, the diameter of afferent, the diameter of afferent is more than efferent artery. Okay, so what will happen? Pressure will be created. Okay, the will be created pressure will be created at the region of glomerulus, and hence blood is filtered blood is filtered into GF. So what is GF? GF is glomerular filtrate. So how glomerular filtrate is formed? Glomerular filtrate is formed when it lacks that protein, which you can say it lacks plasma protein. Why it lacks plasma protein? Because the basement membrane is negatively charged. Plasma membrane is also negatively charged. So here you can admire. So this is negatively charged. So these basement membrane are not allowing the blood corpuscles to get filtered. So GF, of course, they lack plasma protein. So GF lacks plasma protein. Okay, and what does it lacks? It lacks corpuscles. It lacks corpuscles, all varieties of corpuscles like RBC, it lacks WBC, and it lacks platelets. So all the varieties of corpuscles is being lacked by this glomerular filtrate because there is fenestra which is not allowing this corpuscles to get filtered and come into glomerular filtrate. Okay, so how we can say what is glomerular filtrate is glomerular filtrate is blood, okay, minus blood corpuscles which was getting hindered by fenestra and smart, allowing it to get filtered into GF. And of course, blood protein, or you can say plasma protein. So this is how ultrafiltration is occurring, which is of course the first step of physiology of urine formation. Okay, so in physiology of urine formation, we understood about the ultrafiltration thing. So ultrafiltration is like when the pressure, when the diameter of afferent artery is more as compared to efferent artery, blood is getting at the level of glomerulus, the pressure is being created and then blood is filtered into ultrafiltrate. And in ultrafiltrate, we understood about the thing that there is pedicel and podocytes which is forming a slit pore and then blood is getting filtered, but it lacks plasma protein because of the negatively charged basement membrane, and it lacks corpuscles because of the fenestra thing. And this is how glomerular filtrate comprises of blood minus blood corpuscles, and so it is and blood protein. So this is how GF is formed. GF is also called UF or ultrafiltrate, hence because it is formed by the process of ultrafiltration. Okay. I hope you understood what ultrafiltration is. And in next topic, of course, we are going to discuss about the next selective reabsorption thing. Thank you.